thank you for FreeMind to giving us a moment to share the Secretome story. I'm Vinny Jindal. I'm the co-founder, president, and CEO of Secretome Therapeutics. So why do we exist? Well, we observed that for a long time, mesenchymal stem cells and other stem cells have been studied clinically with a couple of very, very clear lessons learned. Number one, they work. Number two, they're extraordinarily safe relative to a lot of other drug classes. And that number three, they're really challenging to manufacture, right? And so the reason is we all kind of start out the same. And then as we age, we become different. So phenotypically, each of us has different stem cells because we've all made different life choices and our genetics have kicked in. And so that donor-to-donor variability creates batch-to-batch variability that has made it really challenging for stem cell-based products to meet the FDA's rigorous manufacturing standards. Some companies and academics have decided to try to solve that problem by going into embryonic or fetal stem cells, which we think comes with a whole other set of baggage, which will make it really challenging to get those products approved. So our reason to exist is that there are about 1,000 surgeries every year on neonates, babies under 30 days old, who have a congenital heart defect that requires life-saving open heart surgery within the first 10 days of life. We get consent from the parents to rescue tissue that's excised from the heart during these surgeries. So in order to cannulate the heart for bypass, about 10 milligrams of the right atrial appendage is excised and thrown away in the operating room. We rescue that tissue, we process it down to the stem cells within that tissue, the cardiac progenitor cells, and we develop that as our medicine, thereby solving the heterogeneity issue with adult cells and the fetal embryonic issue of fetal or embryonic cells. So in addition to being a wonderful substrate for manufacturing, these are actually also the most potent stem cells that have been studied that are non-embryonic because the potency of stem cells is inversely correlated to their donor age. So we've stumbled upon a stem cell that is the most potent, the least variable in its potency, and the least variable in its makeup, making it an optimal substrate for manufacturing. We're a clinical stage company. We're completing right now a phase 1 study in heart failure with preserved ejection fraction. And we'll have the results for that in February. There's an ongoing investigator-sponsored study, speaking to a group that likes non-dilutive capital, where this phase 1 study in dilated cardiomyopathy that has 2 portions to it is fully paid for by a grant from the Marcus Foundation. So this is 12 patients in a phase 1 that are young adults, and then following that, 12 patients in a study of children with dilated cardiomyopathy, all paid for by the grant, where we send study material and we get data. So it's a wonderful arrangement for us. Our North Star in terms of development is to reduce time, cost, and risk to our first FDA approval, right? To be independent of investor capital. And our way of thinking through how to do that is to focus on diseases where you have the 4 elements that turn FDA from a headwind to a tailwind. A disease that is pediatric, lethal, nothing good to treat it, and orphan. So with pharmacology that could address a wide variety of diseases, we chose to study dilated cardiomyopathy and Duchenne muscular dystrophy. Duchenne muscular dystrophy, as the name implies, is a muscular dystrophy, and therefore it's a neuromuscular disorder. However, the leading cause of death is heart failure. What happens in the heart is actually an accelerated version of what happens to all of us. So relative to skeletal muscle, which can rest and relax and repair when we sleep, etc., the cardiac muscle never rests. But inherently, it accumulates injury. So how is that injury dealt with? The cardiac progenitor cells in the myocardium maintain homeostasis through the paracrine secretion of a collection of factors, about 300 factors, collectively known as the secretome, ergo our name. And these cardiac progenitor cells, like all the cells in our body, age over time. And they become less potent at doing their job. Therefore, heart failure tends to show up in our 60s and 70s. When patients with Duchenne muscular dystrophy who have a dystrophin mutation, their muscle presents with numerous micro tears continually, which is why they're wheelchair bound in their early 20s. And so this is equally true of their cardiomyocytes. So the cardiomyocytes whose homeostasis is maintained by these CPCs, because there's injury in every cardiomyocyte, the analogy would be that the cardiac progenitor cells is like a doctor, and they're fixing the cardiomyocytes. But in a patient with Duchenne, their work volume has gone up 1,000-fold, And they themselves are compromised because they also carry the dystrophin mutation. So the whole system breaks down, and this is why heart failure occurs and death occurs due to heart failure in the 20s. So giving back cardiac progenitor cells would be a reasonable therapeutic strategy. And for these boys, it's mostly boys who have Duchenne muscular dystrophy, you could give them back cells from an older donor or a younger donor. In that scenario, you would choose a younger donor. And this is why we're developing neonatal cardiac progenitor cells to treat cardiomyopathy and DMD. There's a clinical precedent. A company called Capricor Therapeutics announced both phase 2 and phase 3 data in the last few years. Their phase 2 data indicated that their drug works. So patients with Duchenne muscular dystrophy have inexorable decline in cardiac function as measured by a loss of left ventricular ejection fraction, or LVEF. You can see natural history, which is pretty robust in this disease, suggests you lose about 2.5 points every year. For 4 years, deramiocel, Capricor's drug, was able to maintain EF. So this is not due to chance. This is real pharmacology. Their drug is a 4-cell construct, and you can see that it includes a cardiac progenitor cell, the adult cardiac progenitor cell. We know that all of this activity is due to the cardiac progenitor cell because we remade deramiocel. This is the phase 3 data that reinforces the finding from phase 2 that left ventricular ejection fraction is preserved relative to placebo. But we've taken the same starting material, the right age-real appendage. We've remade deramiocel in our own lab. Deramiocel is typically about 10% CPC, and we've enriched the CPC content and seen a correlative improvement in ejection fraction. So we're aware the clinical results that they've produced are because of the CPC. So looking at a head-to-head comparison, we took their cell in rodents post-MI whose LVEF is depressed. Deramiocel works. We've seen it work in our own lab, and they've seen it work clinically. It works less well than a pure CPC product, which makes sense. And the adult CPC product works less well than our neonatal product, both at improving left ventricular ejection fraction and alleviating cardiac fibrosis. Getting to the specific properties of our CPC for manufacturing as well, which I led with, this is the state of play for an adult cardiac progenitor cell. 5 different donors. You can see in the component analysis, there's a tremendous amount of heterogeneity. When we looked at 5 different neonatal donors, the exact same cell, the CPC, there's 0 heterogeneity. So the state of play for deramiocel, unfortunately, and any other adult stem cell is that you can only passage them so many times. After 4 passages or 8 doublings, the cardiac progenitor cell from adults loses c-KIT, which is a canonical marker of being a stem cell. And then when you're on to the next donor, after a limited number of doses made, you're in trouble because that donor could be quite different. For our neonatal cells, we can passage them out to 20 with no difference. This just shows 8, but we've passaged them out to 20 and done sampling at 3, 10, and 20 to determine that there's no change in the phenotype or functional activity as measured by improvement in LVEF. We've launched a phase 1 study in heart failure with preserved ejection fraction. It's a simple study. There's 2 doses, 28 days apart. We look at day 56, and there's 2 dose levels, 100 million cells and 200 million cells. We're pleased to see that there have been 0 drug-related adverse events. And we see significant activity. So when we look at the Kansas City Cardiology Questionnaire, which is, a patient-reported outcome, there's a lot of subjectivity to it. Not having placebo-controlled data makes it hard to interpret what really happened. So our way of providing context was looking at the last 2 entrants into the field of HFpEF, the incretins, or GLP-1s, the SGLT2s. And on treatment, in those big phase 3 studies, we saw an improvement about 17 points and the placebo effect's about 10, which gives us some comfort that this treatment effect is real, particularly because half the patients on our study were on both incretins and SGLT2s. So they already got their improvement of 17 points, and we sent them up 20 more. And there's a similar trend that we see for 6-minute walk test. The improvement in kidney function was a surprising finding, but related to the pharmacology. And because we can build new cell products, right? I take my product STEMO1, I introduce a new gene into it, we make that STEMO3, and we can use this clinical signal to guide then where we study our follow-on genetically transduced products. So we're moving forward, having the safety signals from our phase 1 study, clear proof of concept clinically from Capricor, and a clear improvement to Capricor's product in our head-to-head studies on LVEF. in a program with Duchenne muscular dystrophy. I just want to see if this next slide is there. So relative to their study, if we just ran their HOPE-3 study, we would probably produce pretty similar results. But we're not just running their HOPE-3 study. We're running their HOPE-3 study with a more potent drug that we're going to dose twice as frequently in a patient group more likely to respond on therapy and decline on placebo, because we can get patients in an EF range between 45 and 55, where we know that patients under 45 wouldn't respond, and there was a number of those in Capricor's HOPE-3 study. We know that patients over 55 in ejection fraction are unlikely to decline, and we're limiting enrollment to either 55 or 55 plus evidence of cardiac fibrosis, which is also a leading indicator of decline in the next 12 months. So summarizing our programs, we're the only company developing neonatal stem cells, neonatal cardiac progenitor cells specifically. We have 2 composition of matter patents on our product STEM01 and the secretome that comes from it. We've raised $35 million to date. We've produced clinical results, and we're moving into a pivotal study to treat cardiomyopathy and DMD later this year. So, thank you. And I'd be happy to take questions if there are any. Yeah, so that 35 million is our diluted funding figure, but the technology was developed at University of Maryland, Baltimore, off the strength of NSF, NIH funds, as well as the Maryland Stem Cell Research Fund, since it was developed at University of Maryland, and they have granting agencies specifically for stem cell-based products. So, the technology would never have emerged if it weren't for non-dilutive funding. We've additionally won multiple grants from MSCRF as a company. We also won a $400,000 grant from the Friedreich's Ataxia Research Alliance, which is the key patient advocacy group in Friedreich's Ataxia. And with FreeMind, we've applied for, I think 4 more grants, and we're waiting to hear on 2 from them, on 2 of them imminently. So, we think of non-dilutive funding as absolutely critical. The last 4 years of nuclear winter and biotech to fundraise, if you weren't looking at every possible capital source, then woe unto you as a management team. And some of that funding has been absolutely instrumental for us to move forward. Well, thank you.